In the last lecture, we have discussed about the endoplasmic reticulum, and now we are going to discuss about the Golgi apparatus. Golgi apparatus, Golgi complex or Golgi body are the different terms used to describe the Golgi complex. Okay. So these are the manufacturing, packaging and the transport unit of the cell. Golgi complex essentially consists of stacks of membranous sacs. Okay, and these stacks of membranous sacs are called as the cisternae. And several cisternaes are present. Maybe few cisternaes are there or the many cisternaes are there. That is totally depend on the function of the cell and where these Golgi apparatus is present, in which type of cells this Golgi apparatus is present. Depends on that the number of these cisternaes is varies. Okay, diameter of this cisternae varies from 0.5 to 1 μm. So here the thickness and molecular composition of two membranes of the Golgi sac differ from each other. The Golgi sac shows specific orientation in the cell, it having the two sides, each cisternae having the two sides. Here see, we are able to see the cis face and the trans face. Okay, so each cisterna has a forming or the cis face. This cis face is the forming face. Literally, cis means on the same side. Okay, and here cis, from the cis side, whatever the vesicles are there that leave the endoplasmic reticulum and fuse to the Golgi complex from its cis face. Okay, so that's why we call the cis as the incoming transport vesicles are there, which are actually just come here on the cis face. Cis face is called as the, it is the forming or the cis face. Cis is literally means the on the same side, and what I told you that vesicles that leave from the endoplasmic reticulum and fuse to this whole Golgi apparatus or the Golgi complex from this cis face. Okay, and what about the trans face? The trans face is literally meaning is the opposite side. This is the same side. Cis means the same side, and what mean by the trans? Literally, trans means the opposite side. Okay, and from this trans face, the maturing vesicles are present. Okay, so this trans side is called as the maturing phase again. And what happen here? Here the vesicles from this trans phase is leaves the Golgi complex. Okay, so the incoming vesicle, or we can say receiving side of the Golgi complex is called as the cis phase, and the what we can say the leaving side of the Golgi apparatus is called as the trans phase. Okay, so transport vesicles that pinch off from the transitional endoplasmic reticulum merge with the cis phase of the Golgi cisterna and add its content to this lumen. Okay, this explains why the Golgi bodies are usually located near the endoplasmic reticulum. The modified and condensed secretion leave Golgi through trans phase again as the membrane bound vesicles. Okay, so this is the figure from your textbook, And next to this, see cis phase and trans phase of the Golgi apparatus. Okay. Cis phase is the receiving phase of the Golgi apparatus, to which the vesicle from the endoplasmic reticulum empty their content. Okay. This is the convex in shape. This faces the endoplasmic reticulum and receives vesicle from the endoplasmic reticulum, and it receives the immature or unmodified protein. Okay. What about the trans phase? Opposite to the cis phase, the trans phase is the existing phase of the Golgi apparatus, from which the vesicles leaves the Golgi apparatus. It is concave in shape. It faces the cytoplasm. Whatever the vesicles are there, that vesicle exit the Golgi apparatus from the trans phase. Okay. Then it contains the modified protein that leave the Golgi apparatus. Okay. So it about the cis phase and the trans phase of Golgi apparatus. Okay. Here, Golgi body carries out two types of function. One is the modification of the secretion of endoplasmic reticulum and production of own secretion. Cisternae contain the specific enzymes for specific function. Refining of product takes place in an orderly manner. For example, glycolipids and the glycoproteins that are brought from endoplasmic reticulum lose certain sugars and regain others, thus forming a variety of product. Okay, so actually Golgi complex having the secretory function. It secretes different protein, basically sugars and the other substance. Golgi bodies also manufacture their own products. Golgi bodies in many plant cells produce non-cellulose polysaccharides like the pectin also. Manufactured or modified, all products of the Golgi complex leave cisternae from the trans phase of the transport vesicle. While they are leaving from the Golgi, certain markers may get impregnated on their membrane so that they can identify their specific target cell or the cell organelle. So in this way, the Golgi complex are the secretory in function. Okay. So in this way, we have just completed the Golgi complex in this lecture. In the next lecture, we will discuss about the lysosome.